In this class, we discussed about the next instrument PCR. This is the structure of PCR. PCR refers to the polymerase chain reaction. Polymerase chain reaction is an in vitro DNA amplification protocol. It selectively amplifies a specific DNA sequence from any source, that is, virus, bacteria, plant, human, hundreds of millions of times in a matter of hours. The technique is developed by Katie Millis in 1980s. The reaction components of PCR. First one, template DNA contains the sequence of DNA you wanted to amplify. The DNA can be from animals, plants, viruses, or bacteria. And DNA polymerase is an enzyme that makes a new strand of DNA through the sequential addition of nucleotides. And next one, primers. Primers are small segments of single stranded DNA, usually 20 nucleotides long, which bind to a specific region on either side of the target DNA sequence and initiates replication of the target DNA at the point. Primers specify the DNA sequence to be amplified. Next one, buffer. Buffer is a salt solution that helps to stabilize the DNA and other components of the reaction. And PCR is a repeated cycling of three steps, that is, denature DNA. In this step, the DNA is heated to 95 degrees centigrade. This breaks the weak hydrogen bonds that hold DNA strands together in a helix, allowing the strands to separate, creating single stranded DNA. In the first step, that is denature DNA, the DNA is heated to 95 degrees centigrade. This breaks the weak hydrogen bonds. And the next step is primer annealing. In this step, the mixture is cooled to anywhere from 45 to 72 degrees centigrade. This allows the primers to bind to their complementary sequence in the template DNA. And the next step is extension. The reaction is then heated to 72 degrees centigrade. The optimal temperature of for DNA polymerase to act. And DNA polymerase extends the primers, adding nucleotides onto the primer in a sequential manner using the target DNA as the template. And next, go to step 1. Next step. These steps are repeated 20 to 35 times. In PCR, amplification is exponential because for each cycle, the DNA made in the previous cycles can also serve as template. These steps are repeated 20 to 35 times. And the applications of PCR. DNA applications for PCR. It is used to detection of pathogens and diagnosis of specific mutation, and PCR is also used in DNA fingerprinting and detection of specific microorganisms in paternal diagnosis and diagnosis of plant pathogen in paleontology, gene therapy. And the next instrument is gel documentation. This is the structure of gel documentation. Gel documentation. Gel doc, also known as a gel documentation system. Gel image system or gel imagery is a compact and automated gel imaging instrument designed to yield quality images and analyzed results with just the push, push of a button. It is widely used in molecular biology laboratories for the imaging and documentation of nucleic acid and protein suspended within polyacrylamide or agarose gels. These gels are typically stained with ethidium bromide or other fluorophores such as SYBR green. Generally, a gel doc includes an ultraviolet light transilluminator, a hood or a dark room to shield external light sources and protect the user from UV exposure, and a CCD or CMOS camera for image capturing. And the gel dock system consists of cabinet, a digital and a computer. The cabinet is a dark room. The transilluminator that emits light is placed at the bottom of the cabinet. The digital camera is fixed on the roof of the cabinet to capture images of fluorescence or chemiluminescence produced by DNA or RNA proteins after excitation by UV rays. It captures images about 15 to 45 centimeters away from the gel bed. The camera is fully automated that enables the user to catch images of DNA or RNA bands directly from the gels. The camera is converted with the PC by a FireWire port after installing the user interface software. The dock of the cabinet has glass window to view the gel during